Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the lecture 18 and we will discuss about single molecule imaging and tracking. So let me explain you what is single molecule imaging. So single molecule imaging is a method of visualizing and quantifying the dynamics of the biomolecules at single molecule level either in vivo or in vitro. Okay, so here I will play two videos and you can see the single molecules of some protein which we have fluorescently labeled inside the nucleus of a live cell. Okay, so here in this video we have labeled histone H2B with a fluorescent ligand, and you are seeing the single molecules of histones in the cell nucleus, whereas on the left hand side you can see we have labeled transcription factor with a fluorescent dye, and you can see the dynamics of transcription factors. Okay, so single molecule imaging is nothing but it's a method by which you can visualize the individual biomolecules uh, in the live cell or even in vitro also. Okay, so just by looking at these movies, what we can see? So you can see how fast the molecules are moving, or some of the molecules are stably bound. You can see that some of them are not changing the position, whereas some of the diffusing molecules in the cell that you can see. So histone H2B is generally considered as a chromatin bound because it's a histone protein. So you can see some of the molecules are stably bound, but some fraction is also diffusing. So using this method, you can visualize this any protein at the individual molecule level, and you can quantify its dynamics, that what fraction of it is bound molecule, or what fraction, what is the diffusion coefficient, or what is the mean square displacements. Okay. Now, why such kind of method is useful? So you know that several biochemical or microscopy methods that has been used for decades, such as immunofluorescence, Western blotting, chromatin immunoprecipitation, proteomics, or even FREP analysis. Okay, so all these methods are considered as a population-based average measurement methods. Why we call it as a population-based average measurement methods or the ensemble averaging methods? Because you take thousands of cells, you lyse them, and then you get the protein extracts, and that protein extract you use for Western blotting or chromatin immunoprecipitation to quantify that how much protein is there or how much protein is phosphorylated or not. Okay, so it's basically the average. You are making the cell lysate from thousands of cells, and whatever the output result that you will get, that is just the average of so many different cells. But biological systems are highly heterogenic in nature, right? So there are some changes, differences exist between cell to cell. So using this population based average measurement approach, you cannot quantify those kind of cell to cell heterogeneity. Okay, so single molecule imaging visualizes the activity of single molecules and record it with high spatiotemporal resolution. Okay, now, why such kind of method is required for studying biological processes? So you know that the biological processes are highly dynamic in nature. Okay, here I have shown the phenomenon of phosphorylation, dephosphorylation by the blinking of two bulbs. Okay, so you know that the protein kinases phosphorylates some protein, and there are dephosphorylases which removes the phosphate group from the protein. So there is always a cycle between the phosphorylation and dephosphorylation for any particular protein. So let's say in the controlled condition, this phosphorylation dephosphorylation cycle is fast compared to the diseased condition. Okay, and if you want to capture this kind of differences using ensemble averaging method or the population based averaging method such as Western blotting or the mass spectrometry, you cannot capture such dynamic differences, because in both the cases you are taking thousands of cells, you are making the cell lysate, and then if you will do the Western blotting using, and let's say, and phosphospecific antibodies. Okay, so at the time of your cell lysis, let's say 50% cells were having the phosphorylated protein and the 50% cells were having non-phosphorylated protein. So eventually they will not show this kind of differences, but they will show that both have the equal amount of protein or equal amount of phosphorylated protein. Okay, even the mass spectrometry will do the same thing. Okay, but to capture such kind of dynamic differences, you need a method which can provide very high spatio-temporal resolution. Okay, and single molecule imaging is one such method which provides very high spatio-temporal resolution, and you can capture the fast biological processes and measure their dynamics. So what do we see when we are doing the single molecule imaging? Okay, so let's say this is a cell nucleus, and this green color protein is a transcription factor, and this red color site is the specific gene promoter where this transcription factor is supposed to bind for the activation of transcription. Okay, now how this transcription factor reaches to this red color target site? Okay, so it will make this kind of movement inside the nucleus, then it will bind transiently to this kind of blue sites, which are known as non-specific sites, which are not the real binding sites, but it will bind there to make sure that this is not the correct site. Okay, and then it will leave from that position. So it will bind to this kind of non-specific sites, and eventually it will bind to the real target site, and there it will bind, and then it will activate the, let's say, transcription. Okay, so in single molecule imaging, you can visualize this process, like how long this molecule takes to reach to the target site, how many non-specific interactions it makes before reaching to the target site. Okay, and you can quantify the on and off rate of binding of any protein to any target site, and also you can measure the diffusion parameters, that how fast or slow this protein is diffusing inside the cell. You can measure the mean square displacement and diffusion parameters. Okay, so using this approach, what you can measure? Behavior of population of molecules. Okay, so let's say for same protein there are 2 or 3 different type of population of that protein. Let's say one population is involved in one biological function, the other population is involved in other biological functions. So you will get to know that how many different types of populations exist for that particular protein. Then you will get to know the fraction of bound, transiently bound, and unbound molecules. Okay, so as I showed you in the previous movie, you can see this is the histones, single molecule imaging of histones. Now you see that some fraction of molecules are already bound at the target site. Some of them are just diffusing. So you can, the diffusing molecule you can just see for one frame and then they disappear. Okay, so those are the diffusing molecule. So using this method, you can quantify that, let's say out of 100% molecules, let's say 70% molecules are bound molecules and 30% molecules are diffusing molecules. Then jump distances you can calculate. So what is jump distances? So let's say this molecule moved from this position to this position, then it moved from this position to this position. So you can measure that how big jumps it is making. Okay, big or small jumps it is making. If it is bound, it will not make big jumps. If it is unbound, then it will make big jumps. Okay, so here this molecule is unbound, so it is expected to make this kind of big jumps until it reaches to the target site. Once it reaches to the target site, it won't make big jumps. Only a small jumps it will make because of the inherent movement of the chromatin. Okay, then you can also calculate the jump angle. So what is a jump angle? So let's say this molecule in this frame, it moved from this position to this position, then it made the next jump in this direction, Okay, so this is the jump angle. Okay, so whatever it is, maybe 80% or 70%, so that is a jump angle. So you can measure the jump angle that way. So from the jump angle you can get to know whether a protein is moving in one specific direction or it is just a random movement. Okay, it is making jump back and forth randomly. Okay, then you can measure the diffusion coefficient, you can measure the mean square displacement. Okay, now how to observe this kind of single molecules of any protein in live cells? So there are 4 prerequisites for doing single molecule imaging. The first thing is you need a low labeling density using self-labeling halotype. So I'll give you one example. So ACE1 is a transcription factor, and if you do the GFP tagging of ACE1, then you can see the entire nuclear fluorescence, because there are so many thousands of molecules are present in the nucleus for ACE1, and all of them are labeled with GFP. So when you see this cell under the microscope, all the molecules will give fluorescence and you can see complete nuclear fluorescence. Okay, but you cannot see the individual molecules here. So to see the individual molecules, what you need, let's say instead of labeling all the protein molecules present, you just label 3 or 4 of them so that you can see them in the nucleus clearly. So let's say out of so many molecules, if you just label 2 or 3 molecules, okay, then you can see them as an individual molecule, and even you can capture their dynamics. You can track them, that how they are moving inside the nucleus. But if you label so many molecules, then you cannot track the movement of individual molecules. So this is known as a controlled labeling. Okay, you need very low amount of molecules which are labeled, and for this kind of control labeling, you have to use this kind of self-labeling halotag. Okay, so halotag is nothing but it's a small protein moiety of 33 kilodalton. So just like GFP, the way you fuse any protein at the C terminus or N terminus with a GFP, similarly you have to genetically fuse any gene with the at C terminus or N terminus with a halotag, and halotag will make a protein of 33 kilodalton which is non-fluorescent. Okay, so the halotag itself is non-fluorescent, but it binds to the fluorescent dye that is added externally. So let me show you here what is halotag and how this halotag chemistry is used for controlled labeling. So this is your protein of interest, and as I mentioned, it is C-terminally fused with the halotag. So halotag is a non-fluorescent protein moiety. Okay, so the way the GFP fuses with the protein of interest, similarly you can genetically encode this halotag at the C or N terminal of your protein of interest. And this halotag is non-fluorescent, but it can bind to the fluorescent ligand that is added externally. So this is known as the halotag ligand or the halotag substrate. So that ligand binds to this protein with a covalent bond. Okay, so it provides 1:1 protein to dye ratio. Okay, so only one halotag protein can bind to only one dye molecule. Okay, and that is why it is used for single molecule imaging. You can uh, label individual protein with one dye. Okay, so here you can see the representative movie in which these are 3 yeast nuclei are shown. And as I mentioned that in nuclei, you are seeing just 2 or 3 molecules. That doesn't mean that for this protein, only 2 or 3 molecules are present. There are thousands of molecules present inside the nucleus, but you have fluorescently labeled only 2 or 3 molecules so that you can individually see them and you can individually track them, that how they are moving, how fast they are moving, in what direction they are moving. Okay. So this is known as the control labeling that is required for single molecule imaging, and for that you can use the halotag chemistry, and there are some more tags, snap tag and clip tag, which provides the same chemistry for labeling. So you have to fuse your protein of interest at the C terminus with either halotag or snap tag or clip tag. So let's say if you want to do 3 color imaging simultaneously, then protein 1 can be C terminally fused with halotag, protein 2 can be C terminally fused with a snap tag, and protein 3 can be C terminally fused with a clip tag. And for all 3 tags, the fluorescent ligands are available commercially. So those fluorescent dye you have to buy in different colors. Let's say one dye you buy in magenta, other dye you buy in red color, and the third dye you buy in green color. And 3 different proteins using 3 different colored dye, you can track them simultaneously in the same cell. What is next that is required for the single molecule imaging? So highly inclined and laminated optical shade illumination. So we already discussed different types of illumination strategies in one of the previous class. So generally in the epi illumination, you see the entire cell gets illuminated at a time. So all the fluorescent molecules which are above and below the focal plane, they give fluorescence at the same time, and it increases the signal to noise ratio, because the molecules which are out of focus, they give some fluorescent light to your image plane. Okay, then in the turf microscope, you know that the incident light it enters at one particular angle that is higher than the critical angle, so that uh, only a small area, roughly around 100 to 200 nanometer from the cover slip, gets illuminated. But let's say if you want to see some molecules in the nucleus, then turf illumination doesn't work, because it will not illuminate the molecules which are in the nucleus. So there is a slightly modified version of the turf illumination, which is known as a halo illumination, that is highly inclined and laminated optical shade. So in this case, the incident light angle is kept slightly lesser than the critical angle, so that a thin sheet of light passes through the cell, including the nucleus, and it will illuminate only a few molecules in the focal plane, whereas the molecules which are above or below the focal plane, they will not get illuminated. Okay, so why this halo illumination is required? Halo illumination is required to avoid out of focus light from the molecules which are above and below the focal plane. Okay, then the third important thing which is required for the single molecule imaging is the ultra sensitive EMCCD or SCMOS camera, because here whatever the molecules you are seeing, those are the individual dye molecule. So you have to capture the fluorescence coming from individual dye molecules. So it's very weak fluorescence, and you need really a high sensitivity detector like EMCCD camera or the SCMOS camera to capture such low intensity of fluorescence. And the fourth thing that is required for single molecule imaging, that if you are doing it in the yeast cell. So yeast Saccharomyces cerevisiae has some membrane transporter which uh, doesn't allow the entry of this fluorescent dye inside the cell. Okay, so you need to delete them. That is a PDR5 gene in yeast which you need to delete to allow the entry of this fluorescent dye inside the cell. So you can see if you treat the wild type cells with this fluorescent dye, it will not show any fluorescence. But if you delete the PDR5 gene, and then you can see the cells take the fluorescent dye, and you can see some labeled molecules inside the live cell. But this is only specific for yeast. If you are working with the human cell lines, then this method is pretty simple, and you do not need to delete any membrane transporters. This dye can directly go inside the cell human cell lines. And these are the three types of tags which are known as self-labeling enzyme tags, which are available: Halotag, SnapTag, and ClipTag. So these three tags actually ensures 1:1 protein to dye binding ratio. The fluorescent dyes that are generally being used for single molecule imaging and tracking applications are known as the Janelia flow dyes. So just like the Alexa flow dyes, these are the this is a brand name which is known as the Janelia flow dyes. Okay, and just like the other dyes, they are also available in multiple different colors. So you have to select the color based on the availability of the lasers and fluorescence filters in your microscope. Okay, and generally the dyes which are in the higher wavelength like JF646 or JF635 are the most preferred dyes because they induce less phototoxicity because of the far red range. Okay, so you can choose any of these colors, but preferably you should go for the longer wavelength like JF646 or JF635 or JF585. And all these dyes, you can see they are ideal for single molecule imaging, super resolution imaging, confocal imaging, intracellular labeling, any analytical method that relies on fluorescent labeling and detection. And now they are commercially available. And this dye have the advantage of over the other dyes like the Alexa flow dyes or so they have a high quantum yield, and their fluorescent lifetime is longer, because for single molecule imaging we really need dye by which we can observe the molecule for longer time. So if you use a dye which can be photo bleached easily, then those dyes are not good for single molecule imaging, and you need a dye which shows good photo stability. So here is the workflow for the identification and tracking of single molecules. So how the single molecule imaging and tracking is being carried out. So see, this is the raw time-lapse movie. You first label your fluorescent, you first label your protein of interest with the Halotag dye or the snap tag or the clip tag dye as we discussed before. And because of that, you will be able to see the single molecules like this. Now you want to image these molecules for longer time. So you will try to illuminate it with the lowest laser power possible. Okay, so that you can see the molecules for a long time without photo bleaching. So your raw images will be little noisy. As you can see here, you can see the single molecules here, but there is a huge a lot of noise in the image, right? So we used computational filtering method, which is known as the bandpass filtering, to remove this noise. So after filtering the image, you can see now the single molecules are easily detectable, and the software can detect them uh, yeah, robustly using intensity-based thresholding. Okay, so generally this kind of tracking, we use MATLAB-based tracking software. So those softwares recognize these particles in all the frames of the time-lapse movie using intensity-based thresholding. Okay, so once the particles are detected, the next step is to connect these particles from one frame to the next frame to make the trajectory. Okay, so first thing is it detects the particles in all the frames. So let's say suppose if you capture a time-lapse movie of 1,000 frames. Okay, so then the software will connect this molecule from this frame to this frame, from this frame to this frame. Okay, so to generate the trajectory. And once the trajectory is made, we have to define whether this trajectory is for the bound molecule or the unbound molecule. So how you can define that whether the trajectory is for bound molecule or unbound molecule? So if your protein or this spot moves lesser than this distance of 0.35 micrometer, okay, that we call as a R mean distance. Okay, so if your molecules moves lesser than this distance frame to frame, that means it is a bound molecule. And if it moves more greater than this distance like this, then you should consider it as an unbound molecule. So how we determine this distance? So for that, we did the single molecule tracking of chromatin bound histones. And we observed that the histone proteins, if you track, they do not move more than this distance, okay, frame to frame. So that means we can consider this distance as a to determine whether the molecule is bound or unbound, because the histones, which are already known to be chromatin bound, they do not move more than this distance. So basically, if the molecule makes smaller jumps like this, frame to frame, then it is a bound molecule. If it is a free molecule or the unbound molecule, it makes larger jumps, okay, like this. And sometimes you may get hybrid tracks. So initially the molecule was unbound, it was making bigger jumps, but then it was bound for few frames, and then again it started moving on. Okay, so again it started making big jumps. So this is known as the hybrid track, in which in the initial positions and the end position the molecule was unbound, but for some time the molecule remained bound. So we use several kinetic modeling to determine whether the molecule is bound or unbound. And here you can see the X and T representation of the trajectories. So this is known as the kymograph. Okay. So here, let's say I took the time-lapse movie for 300 frames. So on the x-axis, from 0 frame to 300 frames are there. And this is the XY projection. So you can see how long this individual molecule survived. So let's say this first line represents this molecule. Okay, so that molecule survived from zero frame to, let's say, somewhere around 140 frames. Okay, whereas the other two molecules, like let's say these two molecules, they survived from the first frame to the last frame. So for all 300 frames, you could see that molecule, whereas this molecule, you could see from first frame to roughly around 140 frames. Okay, so this is the residence time, that how long this molecule survive. Okay, so you will capture this data for thousands of tracks, and then you will take the mean residence time, that how long these tracks are surviving. Okay, and then that is known as the survival probability distribution. So you will measure the residence time of each of this track, and then you will plot it like this, which is known as the survival probability distribution. And sorry, and from this you can see if this survival probability distribution fits well with the double exponential decay curve. So that indicates there are two types of bound molecules. The blue fraction is known as the specifically bound molecules. So these are the molecules which are actually at the target site, and the orange fraction represents the non-specifically bound molecules. So these are the molecules which are not at the target site, but they are just making transient interactions with some chromatin or some other proteins in search of the target site. Okay, and the gray fraction represents the diffusing molecule. So from the residence time analysis, you can find out the type of bound molecules, the specifically bound molecules and the non-specifically bound molecules. You can also plot the MSD curves, mean square displacement curve, against time, and based on the shape of this curve, you can say that whether this protein showed confined diffusion or the anomalous diffusion or the Brownian movement or the directed diffusion. Okay. So generally, if you are tracking the molecules inside the cell or inside the nucleus, it will show confined diffusion because the movement of the molecule is confined within the nucleus or within the cell. Whereas if you track any molecule in vitro system, then they may show directed movement or the Brownian movement or the anomalous diffusion. And here you can see the log D histogram. So D stands for the diffusion coefficient. Okay, so you see this is the curve for the chromatin bound histone H2B, and this is for the free halogen. So this is the dynamic range of the diffusion coefficients. If it's a bound molecule, its diffusion coefficient is within this range. But if it is a free molecule, its diffusion coefficient is within this range. And for any protein, if you track the diffusion coefficient, you can see different population. Let's say for this TBP, this is a Tata binding protein. Okay, so there are three population of that protein molecule present. One is tightly bound, the other one is intermediate bound, and the third one is the diffusing molecule. So maybe these two population, tightly bound and intermediate bound, so they may have two different biological functions. Okay, and that's why that protein showed total three population. But let's say for this protein, polymerase 2 and TF2D, there are only two population exist. Okay, the first population is the bound molecule and the other population is the unbound molecule. So for TF2D, most of the fraction is unbound and only a small fraction is bound. Whereas for polymerase 2, mostly both the fraction, bound and unbound, they are of equal size. Okay, so you can, from the diffusion coefficient analysis, you can get to know how many types of population of that protein exist. So this data we have taken from the Nguyen et al. 2021, where they have used the single molecule imaging and tracking to track the individual component of the transcription pre-initiation complex. Now coming to the imaging strategies. So for single molecule tracking, you have to take the time-lapse movies, right? Because you want to capture that how the molecules are moving, okay, from one frame to the next frame. So what time interval you will keep between the frames? Okay, so we capture this data using two strategies. We capture images with fast imaging regime and the slow imaging regime. So fast imaging regime means we want to see the molecule continuously. Okay, so there is no interval in between. Okay, so only the camera processing time is the interval. So let's say camera itself takes 15 milliseconds to process one image. Okay, so here our time interval is just 10 to 15 milliseconds. That is actually not the interval, but it is actually the camera processing time. Okay, so we are continuously observing the molecule by keeping the lasers on, keeping the camera on continuously, just like a continuous video recording that we do. Okay, but the drawback of this approach is that we will bleach the molecule faster because we are continuously imaging, right? We are continuously keeping the lasers on, we are continuously keeping the camera on. So molecule will bleach faster. So if I want to measure the residence time, that how long this molecule stays at that position, then the residence time will be compromised because of the photo bleaching. Okay, so this is not the good approach to quantify the residence time. But from these kind of movies, we calculate the mean square displacement, diffusion coefficient, and fractions of bound and unbound molecule. So all the diffusion related parameters, the diffusion dynamics, we can capture or quantify from these movies, but not the residence time. But for quantifying the residence time, we have to image the cell at different time intervals, like 200 millisecond time interval or 1000 millisecond time interval, so that with this time interval, we can image the molecule for longer time before it photo bleaches. Okay, and that gives us the real estimation of the residence time. Um, and so basically, to capture the full dynamic information of any protein, you should image it with two imaging regimes: fast imaging regime and the slow imaging regime. And depending on how strongly your protein is bound, you have to decide the imaging time interval. Let's say if you are if you are imaging a protein with a residence time of around 8 to 15 seconds, then maybe this 200 millisecond interval is good. But let's say your protein is very tightly bound, and its residence time is in the range of, let's say, 1 minute or 2 minutes. Then in that case, you should go for the higher residence, higher interval imaging. Okay, so that thing you need to optimize based on your protein of interest. So here I have summarized the overall or the broad applications of single molecule imaging. So these days, single molecule imaging has a variety of applications in to address variety of biological questions. Okay, you can use single molecule imaging to study the protein conformational dynamics using single molecule FRET. You can use it for ion channel dynamics using single molecule FRET. Then you can use this single molecule tracking that we discussed for studying any DNA protein or RNA protein or protein-protein interactions in live cells or even in fixed cells or even in vitro systems. Then it has several applications in the field of virology. If you want to see how the single viral or virus particle infect the host cell, then you can use the single molecule imaging method to capture such kind of dynamics. Then to study enzyme dynamics, like if you want to study the dynamics of topoisomerases, helicases, kinases, phosphatases, all these are DNA interacting proteins, and how they actually work on DNA, it is still in its infancy. So that's why this technique is very powerful to quantify the time scale of the biological process. You can literally quantify that how long this topoisomerase takes to for the removing the supercoiling of the DNA, okay, or how long this kinase takes to phosphorylate any particular protein. Then it has several applications in signal transduction, and also it can be used for studying transcription dynamics. So how long the transcription factor binds to the gene promoter for the activation of transcription. So those kind of questions you can address using single molecule imaging and tracking method. And also it is quite versatile because you can use this method in human cells, in yeast, in bacteria, or even in mouse embryo. So it is compatible with several model systems, and that's why it is quite versatile, and you can use it for many different applications. So now I will show you some specific examples that what kind of new information can be revealed using the single molecule imaging method. So here I have shown some data from this paper, Mehta et al. 2018, and here we studied how the chromatin remodeler RSA2 affects the dynamics of a transcription factor. So what I have done, like I have tracked the single molecules of the transcription factor ACE1. So ACE1 is a transcription factor. I labeled it with hello tag and using the JF646, the fluorescent dye. Okay, so I could visualize the single molecules of ACE1 in the cell nucleus. And then I measured its residence time in the presence and absence of the chromatin remodeler RSA2. And what you can see in the absence of RSA2, the blue fraction, which represents the specifically bound molecules of transcription factor, which are actually activating the transcription. Okay, it has reduced from 10% to 3.8%. So that means this chromatin remodeler is important for the recruitment of these transcription factors at the gene promoters. Here we have tracked this transcription factors specifically at the gene promoter. Okay, so you can see that this chromatin remodeler is important for recruitment of this transcription factor at the gene promoter. Otherwise, in its absence, the fraction of specifically bound molecule reduces. Interestingly, what we observed, we measure the residence time, and what we observed that in the absence of chromatin remodeler, the residence time has increased from 2.18 seconds to 5.2 seconds. Okay, so here the small fraction is binding, but it is binding with a long residence time. Okay, so these molecules are staying there for longer time compared to these molecules. So there is a simple equation by which we can actually calculate the search time. So what is the search time? The search time is the time that a transcription factor takes to reach to the gene promoter. Okay, so we quantified using this equation, we quantified the search time for this transcription factor ACE1 to bind to the gene promoters in the presence and absence of RSA2. And what we observed that when the chromatin remodeler is present, the transcription factor just take 8 seconds to reach to the gene promoter, whereas in the absence of chromatin remodeler, it takes 25 seconds to reach to the gene promoters. So just to give you an idea that this kind of, you can, using this method, you can actually quantify the time scale of the biological processes. Okay. Then it has a wide variety of applications in the field of virology to study various events of viral entry into mammalian cell. So you can use the single molecule imaging for, let's say, single virus particle tracking, that how viral particle finds its receptor on the cell surface. Then protein, to study the protein membrane interaction, that how a viral particle undergo membrane fusion. Protein nucleic acid interaction. This is known as protein induced fluorescence and fluorescent enhancement and FRET assay. Then viral helicase activity measurement using SMFRET. And also you can study the viral assembly, packaging and architecture using a variety of single molecule imaging approaches. Here I have cited a very nice review on this, that is Banerjee et al. 2018. If you are interested in knowing the broad applications of single molecule imaging to study various aspects of virology, please read this review article and see how they have shown different data and how different technologies have been used to learn more about the biology of viruses. Then this is another applications of single molecule imaging, which is known as single molecule FRET. So single molecule FRET is used to quantify the protein conformational dynamics. Okay, so what is a protein conformational dynamics? So let's say this is one protein. Okay, and in here it is in the open state. Okay, but when it binds to a ligand, this orange color molecule is the ligand, then it is converted into the closed conformation. Okay, so if you want to quantify that how fast this process happens, like the protein remains open or it remains in the closed conformation. Okay, so you just label two ends of this protein using uh, FRET dyes. One is with the, let's say, red color fluorescent dye and other one with the green color dye, and generally this dyes should be a good FRET pair. Okay, generally people use Psi3 and Psi5 for FRET analysis. Okay, so so when this protein conformation closes, this dye molecule come in close proximity to each other, and that gives enhancement of the FRET signal. Okay, so you can actually measure the FRET and you can appreciate the time scale. Within few seconds, this protein goes on and off, okay, for so many times. So the biological processes are so fast, and to study to reveal the dynamics, you need such kind of methods which are so fast and they can quantify this kind of fast dynamics of protein conformational changes. Okay. So single molecule FRET is specifically used to study the protein conformational dynamics. Then here another study using single molecule imaging and tracking. So in the live cells, these authors have tracked the dynamics of telomerase. So you know that telomerase is very important enzyme in the cell, and it is actually required for maintaining the length of the chromosomes, and it has very important role in aging because it regulates the length of the chromosomes so that the aging is uh, so the so that the cell will not age faster. Okay, so in this paper, authors have studied the dynamics of this protein, the telomerase, at different locations. Now, this telomerase localizes to different places. It localizes to telomeres. It also localizes to the Kagel bodies, and it is also moving in the nucleus. Okay, so at three different locations, they tracked the movement of this protein, and what they found that at three different locations, this protein, the telomerase, shows different diffusion dynamics. So in the nucleus, it shows three population. Okay, uh, blue, which is represented by blue, red, and green. Okay, that indicates two or three different biological functions. Whereas in causal bodies, it only shows two types of diffusion dynamics, and in the at the telomeres also it shows only uh, mostly the two fraction population, but there is a small third population also exists. Okay, so yet it is not known that what are the different biological functions of these different populations, but it is possible that all these populations have different biological functions. Okay, so this kind of diffusion dynamics you can study for any protein of your interest. So coming to the current limitations of single molecule imaging and tracking. So the one of the major limitation of single molecule imaging and tracking is the photo bleaching of the fluorescent dyes. Okay, so still the dyes that we discussed, the Genella flow dyes, they are good, they are quite photostable, but still they are bleachable. Okay, so for long imaging applications, or if you image them for a long time, they will not survive. So still photo bleaching is currently one of the most important challenge in single molecule imaging and tracking. And most of the current single molecule imaging and methods observe the molecules in the single focal plane. So as I mentioned that here we take the time lapse movies. Okay, now we really want to go fast at every 15 millisecond or every 200 millisecond. So we cannot go for capturing the entire Z stack. Okay, because Z stack capturing itself takes, let's say, 15 seconds. Okay, even if you have the very fast microscope, taking the Z stack of a cell will take at least 10 seconds. And here you want to go as fast as like 15 milliseconds or even 200 milliseconds or 1000 milliseconds. Okay, so within if you cannot, for single molecule imaging you cannot go for taking the Z stacks. What you have to do is just you have to observe the single focal plane. So what will happen, the bound molecules in this focal plane, they will remain bound, they are not going to change the position. Only the diffusing molecules will come in this focal plane and they will come in and go out. Okay, so what we are seeing in single molecule imaging, by imaging single focal plane, we are observing the bound molecules, okay, which are not going to change the focal plane, but the diffusing molecules will keep changing the focal plane. So if we can do this thing in 3 dimension for covering the entire cell volume, then it will be very advantageous. Then we have the limited labeling strategies for controlled labeling of mRNA, lipids, and carbohydrates. So currently we have a good number of methods to uh, for controlled labeling of proteins using this halo tag or snap tag or clip tag, right? But for if you want to see the single molecules of mRNA, lipids, carbohydrates, you have a very limited or even no labeling strategy. For mRNA, there are some ways to visualize the single molecules, but for lipids and carbohydrates, we have limited options. And also for single molecule tracking, you need the automated software with machine learning for particle detection, tracking, and data analysis. So currently, a lot of manual efforts are required for tracking the molecules from the time-lapse movies, but some kind of automation or the machine learning based automation is required so that the software can automatically detect and track the particles for several frames. So with this, I would like to end this class, and our next class we will discuss about optical tweezers and traction force microscopy. Thank you.